Okay, so I'd like to start today's lecture organized by the Keio University Global Research Institute, which just opened last year, and I'm a vice director there. So we try to facilitate our global activity by this institution. And we just started a lecture series of this KGRI, Keio Global Research Institute. So you are the first speaker, actually, of this lecture series. And so I'm very glad to have Professor Per Uhlén from Karolinska Institute, Department of Medical Biochemistry and Biophysics. Actually, Per and I have the common PhD mentor, who is the Anita Peria. Actually, she visited here 2 weeks ago and gave a lecture, so you remember very well. Although we didn't overlap each other, but our PhD mentor is Anita Peria, and that's why I got to know her. And we've been collaborating for more than 10 years, about 10 years, and he kindly accepted distinguished guest professor of Keio University, appointed this year. Actually, my student Simon, he spent a couple of weeks in Per's lab to learn the new techniques. And Per, he graduated from the Royal Academy of Institute of Technology in Stockholm, KTH. Then moved to the Karolinska Institute as a PhD candidate and got a PhD there. And he's been working on the calcium imaging. He's expert on the live imaging of calcium and other ions and etc. and interested in both the cancer biology and neuroscience. Yeah. And so today, the title of his talk is Structural and Functional Abnormalities in Cancer and Psychiatric Diseases. So please first start your presentation.
Okay, thank you. Thank you very much. Yeah, first of all, thank you for the introduction, and I'm honored to be the number one of this seminar series then. And I'm very happy that I was invited to come here to Keio Medical School and to give this lecture that is going to be called Structural and Functional Abnormalities in Cancer and Psychiatric Diseases. And this is quite a broad title, and this is because my lab, we are working both with, one could say, cancer and also with neuroscience. And therefore, we're doing. Therefore, this title is also about both cancer and psychiatric disease. And I will tell you today about two stories. One project that was recently published that is called Whole Tissue Biopsy Phenotyping of Three-Dimensional Tumors Reveals Patterns of Cancer Heterogeneity. And this story will be, I will tell you about the development of something that we call the DIPCO pipeline and its clinical application. And then I will also tell you another story about a project that we will soon submit that is called the Mood Disorder Susceptibility Gene CACNA1C Modulates Spontaneous Activity in Progenitors of the Developing Brain. And this is a multidisciplinary story spanning from mathematical modeling to behavior tests. And as Masato already told you, my background is actually in physics. So I started as an undergraduate in the Royal Institute of Technology in Stockholm studying nuclear physics. And then I got interested in medicine through nuclear imaging, medical imaging that is used in hospitals and so on. And I was a PhD student in Astrid Lindgren Children's Hospital and had the same mentor as Masato, Anita Peria. But after then, I've been mostly working with cell biology and medicine and not so much nuclear physics anymore. So this first story that I will tell you about is that we also have a lot of collaboration with Keio actually, Keio University. And this is Nobuo Yuki and this is Shigeaki, that both come from Keio University. And they are postdocs in my lab, and they are also co-sharing this first authors of this paper that was recently accepted in Nature Biomedical Engineering. And actually this week we also learned that it was selected as a cover illustration of this month issue of this journal. So we were very happy about that. First I will tell you a little bit about the background, about some techniques that we are using in this, this paper and this project. And one essential technique is tissue clearing. And I guess that many of you already know about this, but just very briefly, what we do with tissue clearing is that we make tissue transparent, as you can see in this picture. Here you have a mouse brain, and then after this clearing process, you can see that you can see right through it. So the brain is still here, but it becomes transparent to light. And that is, of course, strength when you are using microscopy to try to visualize things within the brain. So the light can penetrate much easier through transparent tissue. But this is actually a quite old technique. It was first introduced in 1911, actually. But just recently, when fast computers and very good microscopes are being developed, this technique has kind of been very popular again, and many more protocols have been developed to make tissue clears, and some of them are here called BUB, clarity, cubic, iDisco, scale. I think both cubic and scale come from Japan. There are different ways. They are optimized for different things. If you want to do immunostaining, or if you want to do this with GFP and transgenic animals, and so on and so forth. And in our paper, we have used this iDisco quite a lot. So another technique that is very important for us is this light sheet microscopy. And with light sheet microscopy, it's a little bit special because we have an object here, which is this cylinder here, and then you have two objectives that are perpendicular to each other. So you have, this is the objective that is used for the excitation light, for the light that is used to excite antibodies and fluorescent tags. And it looks like this. It has a sheet, and then the strength with this is that you can put the camera here at the back side of this objective, and then you can record an entire frame. And this is very important when you are doing, like we are doing, these 3D stacks. It takes so long time to scan through one tissue. It can take several days. And these experiments are then very time consuming. They produce a lot of data, and terabytes of data. And of course, if it takes several days, if something goes wrong, you know, somebody opens the door of this room, the whole experiment could be ruined, and so on. But with this new microscope, it's much faster then. And yeah, here is a movie showing this in action. So you have this objective here, and then you can see a lens there, and that is this objective. So there is a camera behind this objective, and then you can record entire frames. And this goes much faster. So today, the latest technology is actually not a sheet of light, it's rather a beam, but the beam goes so fast, so it could be used as a light sheet, yeah, to excite in a whole sheet. Another thing that is very central to this first study that I will tell you about is this intra-tumor heterogeneity. And this means that cancers are actually assembled of heterogeneous populations of cells, meaning that the cells are very different. They are not homogeneous. It's not the same type of cell everywhere. It's just a mixture of a lot of different cells. And this is very important when treating cancers, of course, you know, to know about this heterogeneity. Things that we will tell a little bit more about that we have looked into is this EMT, which stands for epithelial to mesenchymal transition, and also angiogenesis, this the vasculature of tumors are also very important and can tell how aggressive and how how aggressive the tumor is. So it's an important thing to study, both of these processes. And EMT, this stands for actually when epithelial cells in a tumor are stick together. This is very important for a tumor, and when the cells starts to dissociate, meaning that the cells are losing their attraction to each other, one could say, and then the cell can start to flow in the body and make metastases and so on. And that is a very bad thing, and that is also very important to know when you are diagnosing cancers, of course. And to study EMT, one can study certain proteins. And what we have been interested in this study is E-cadherin. And this is a small protein in between the cells that fuse cells together. And then there is another one called N-cadherin, that if that one is upregulated, then it means that the cells start to dissociate from each other. And also vimentin is a protein that is important for this dissociation. And CD34 is a marker of blood vessels that we have studied. So first what we wanted to do, because in this work we were interested to use this method that has been applied a lot in basic research labs all over the world with this clarity or clearing process, but it has not been applied on clinical samples. So the two guys that I showed you before, who are the first authors of this paper, Nobuyuki, he is a urologist from Keio University Hospital, so we came up with this idea that couldn't this method be used to study human samples? So since many of the cancer specimens that are used for pathologists are stored in this paraffin embedded format, it's called FFPE, which stands for formalin fixed paraffin embedded samples, and these are stored in biobanks all over the world. So first what we wanted to do was to see if we could remove this paraffin from the samples and then apply this clearing process and see if that was a problem that could disturb the experiment. So this cartoon illustrates this. So in the beginning we took a sample from a mouse. So we took a sample and then we embedded it in paraffin. And then we removed the paraffin again, and then we did this clearing process. And then we visualized it with a light sheet microscope. And then we compared it with not doing this paraffin process. And we wanted to see if it was if there was any difference between these samples, if this paraffin process destroyed the samples somehow. So you see in this experiment, we have only formalin fixed, and this is this path then. And then the DIPCO is what we call this process. So this is with the paraffin, and then you can see that they look very much similar. So it seems like it was no problem doing like this. The paraffin did not destroy the samples in any way. Next, we used real cancer samples from humans. And what we were interested in was to study these EMT markers that I told you about, this E-cadherin and N-cadherin. And you see here is a sample. Maybe you can we lower the light a little bit? Or? because it's a lot of images and maybe it could be seen a little bit better. Yeah. So you see here is the tumor then, the entire tumor. And then we have stained it for E-cadherin. And Yopro, it's a staining only for the nucleus of the cell. And then here is like if we do a projection and just look at the 2D image, you can see the Yopro that stains almost all cells homogeneously. And this is the way it should be, that because all cells have nucleus, right? And then E-cadherin here shows up, at least in the paper, you can see a little bit here, red. And then if we do pseudo staining of the E-cadherin, you see that it's more inhomogeneous, meaning that here it's stronger staining compared to there. And this is an indication then that you have this EMT process going on and that the tumor is heterogeneous. If it would be homogeneous, of course, then the staining here would be the same all over. But it's varying. Here it's strong, here it's weak. And this is important information for pathologists when they are going to stage tumors and tell what kind of treatment plan the hospital should provide. And if we zoom into here, you see the Yopro again is quite homogeneous, but the E-cadherin is varying. And you have regions where some cells are very strong and some cells are not strong and so on. And this is typical for this heterogeneous tissue. This image here just shows how the tumor becomes transparent when we use this iDisco protocol. And the same also goes for this N-cadherin then, which was this other that is supposed to go up then in this tissue. And you can also see that you have here an inhomogeneous pattern of this N-cadherin staining, telling us that this tumor is heterogeneous. Here is some movie also showing then the tumor in 3D. And we spin it around, and the first one here is with E-cadherin then. And you see that it has some regions where it's stronger and some where it's weaker. And if we do this pseudo scanning, pseudo coloring, you see that there are regions and also inside of the tumor where it's stronger staining. We can zoom in and really go into a tumor and analyze and look into what is going on inside of the tumor. And here is the same, another movie for N-cadherin, where N-cadherin is colored in red, and Yopro, this nuclear staining, is colored in green. And we can scan through, and this also makes it possible for pathologists to make like a digital diagnose of a tumor. You can scan the entire tumor, and then they can, sit at the computer and analyze it and look into it. Today, they are spending a lot of time at the microscope and looking into it. Yeah, you see these different regions where you have more or less. And also, we use this CD34, which is staining for blood vessels. And using that one, we could also see that, some vessels here, we could actually measure the thickness of the vessel. And you, with this picture here, you see that red vessels are thicker. And green and blue is thinner. So, and this is also very typical for a tumor, that you have a varying thickness of the blood vessels. And the more this is varying, the more aggressive the tumor is. So it's a way of measuring how severe the cancer is. So we try to Yeah, here's an image or a movie showing this, that CD34 is stained in red, and it stains blood vessels then. And with the computer you can kind of digitally create blood vessels and you know, make them in a digital form. So we can study them in detail. And the red one here, then you have thick regions of the blood vessels, whereas the blue here are thinner. And we could also go in and count single cells of a tumor, the number of cells, and analyze them cell by cell with different stainings. And in this one, in this picture here, we looked at vimentin. So this is a tumor then that we counted to have 30,214,289 cells inside of this tumor. And we stained then vimentin in green. And then we could, in each cell, measure the vimentin expression. And here is a movie showing then each single cell here. You see, all these dots here are single cells. And you see again here that some of them are very reddish areas, and some are blue and green. Okay, another important thing when it comes to clinical samples is that it's something the pathologists don't want to give away the samples. You know, some of them could be very precious. It could come from a very rare disease or something. So, and since what is usually happening when you ask a pathologist for a sample is that they pick up their block of cancer tissue, and then maybe they cut 1 or 2 slices of that and give to the basic researcher. And then, but they want to keep their block of tissue. So one thing that we explored in this project was the possibility of re-embed the sample in this paraffin that we removed in the beginning, and then to be able to give it back to the pathologist so that they can put it into the biobank again for future experiments, or if they need it in the future. So here you see that what we did was that before doing this clearing and imaging process, the tissue looked like this. So here we use this hematoxylin and eosin staining, and then we also use another marker, Ki67, and stain just to look for the pattern before doing this examination. And then we did the examination, and then we put them back into paraffin again, and then we again examined them in a similar way. And then you can see that these are almost similar before and after. So we didn't destroy the samples with this method. And we also used a number of other antigens to look into to see that we really didn't destroy the samples. So you see here Ki67, as I showed you, P21, P27, P53, and so on and so forth. Many different antigens. And then we looked at a number of tumors. I think, was it 8, 1, 2, 3, 4, 5, 6, 7, 8 samples, 8 different tumors. And next, we also wanted to see if this could actually be applied clinically to help staging tumors. And you see here is a picture of a bladder, and it's staged, in different cis. You have TA, you have T1, T2 to T4, and this is how much muscle invasive the tumor becomes. And this is a very important information for physicians, because of course if it's very superficial, they can remove it with surgery, this tumor, and then do an immuno treatment like this BCG or something, and that is a very successful treatment of bladder cancer if it's superficial. But if it's muscle invasive, like here, T3, T4 and so on, then that method cannot be applied. Then what usually the urologists are doing is that they remove the whole bladder. And then in some cases there is a gray zone where it's very difficult to say if it's muscle invasive or not. And of course, if they say that it's muscle invasive, even though it's not, that will mean that the patient has to remove their bladder. And that is a lot of suffer, of course, for the patients. And the opposite, if they don't say that it's muscle invasive, but still it is. They do this first treatment, they just remove this superficial, but there are still cancer left in the muscle tissue. And then maybe that could lead to death or something if that is not treated correctly. So we targeted, we tested our method to see if we could more accurately actually stage the tumors if we used the 3D method. So we used something here that called a ROC analysis, or ROC analysis, I should say, which stands for receiver operating characteristics. And I just briefly want to say that what this ROC curve is doing is that we are trying to say how accurate a method is to determine what you are measuring. And this curve here is showing the strength of the method. And if it goes up here and to the side here, then it's a perfect method. And if it's just a straight line like this, then it's more like a random method, like, you know, just a random. So we use this analysis in order to evaluate our method and to see if we could stage tumors more accurately. And we also measured something called vessel tortuosity and then kurtosis and skewness. And these are just statistical factors or way of characterizing values when you're doing statistical testing. And doing this, we were able to actually score quite well when it came to staging. And then you see that we divided like normal tissue, this is not any cancer, and then less than stage T2 or more than stage T2 and less than stage 2. And then we measured all these values that I told you about, the skewness and tortuosity and so on. And we looked for the parameters that had this statistical significance. And one was vessel radius. And if you remember from this movie where I showed you red vessels and green and so on, and red are very thick and blue are very thin. So we measured actually all the radius of the vessels within the tumor. And putting this together, this was a rather long, tedious process, of course, but in the end we came up, if we combined the vessel radius and the kurtosis, we got the best result when it came to this ROC analysis. And you see that this curve is looking quite nice here. And the value for that one was 0.839, which is actually very good. And if you compare with, many of you are familiar with this PSA test, for instance, for prostate cancer. And I think that value is 0.65 or something in accuracy. So this showed that this method could improve accuracy when it comes to staging tumors. And we also looked at ovarian cancer, and in this case, then we were interested in to see if they were platinum sensitive or resistant, which is also, of course, very important to know maybe from the beginning before you start a treatment, to see if this patient will actually respond to the treatment or not. To cut a long story short here, we were able to show that this method was also, the DIPCO method was good in predicting survival, and also if the patient were sensitive or not to the treatment. And here you can see 3D images of ovarian cancer. Here's a platinum sensitive, and these are vessels again stained. And you see that then they are more bluish. They are a little bit thinner vessels and more homogeneous. If you compare to this platinum resistant, you have more thicker vessels, and you have also more heterogeneous pattern. It's both red, green, and blue, while here it's more bluish, and homogeneous. Okay, so just to conclude this first part of the story. Hi, Okano. Hi. I told you about this DIPCO method that we named this DIPCO pipeline. It stands for Diagnosing Immunolabeled Paraffin-Embedded Cleared Organs. And we just made this figure like this because it's a way of like reusing samples. You know, we do clearing first of the sample, then we do imaging, and then we do re-embedding, and then we can put it back into the biobank again. So this brings me to the next part of this talk, which is about an ongoing or almost done project. We are just in the end here of running the final experiments before submission. And the title of this one is the mood disorder susceptibility gene CACNA1C that modulates spontaneous activity in progenitors of the developing brain. And the first author, Erik Smeder, was a PhD student in my lab that just recently defended his thesis. And this has been also part of my lab to look at stem cells, and we have worked with this for more than 10 years, and we have a number of publications on this matter. But this project then, we aimed on this CACNA1C gene. And what we did, the start of this was actually that we took stem cells that were proliferating, and then we started to differentiate them towards to becoming neurons. And then they go through these different steps of differentiation. After 4 days, and then after 8 days, they start to become neural progenitors. And then after 16 days approximately, then they become neurons. And we were interested in, since my long-lasting interest in research has been calcium signaling, we were interested in seeing how calcium was varying through this differentiation process. And what we were able to see was that in the beginning, the cells were actually not so active when it comes to calcium activity. So these are stem cells that has been differentiated towards to become neurons, and then we load them with this calcium probe, which is called fluo3, and then we look at them in the microscope. And then you can see that there is a lot of activity going on here. They are blinking, and they have a lot of activity. And we were interested in seeing what genes or proteins were actually responsible of starting this process. So we performed a lot of experiments on this. And one experiment where we made a PCR array with 90 genes, and these genes were involved in neuroscience related, ion channels and transporters. And since we knew that at day 4 we had very little activity of these cells, and then at day 8 we have a lot of activity in these cells. This panel here shows that. So what we did then was that we ran this PCR array first at day 4 and then at day 8. And then we compared the difference between each of these 90 genes at the different time points. And then we saw that some genes stood out to be very there has been a lot of change in these genes during this differentiation. And one of those were this CACNA1C. CACNA1C, we also published a paper, also I think in 2017, where we made a database where you can look into genes and see how they are related to medical diseases. And this is the address to this database that we did. And we focused this database on calcium signaling genes. So it should be, the database is built up of genes that are involved in calcium signaling. So it could be, you know, transporters, or it could also be kinases like CAM kinase 2, which are sensitive to calcium. And if one then looks at this gene CACNA1C, you see that you get a lot of diseases here. And it's autistic disorder, it's depression, and yeah, many diseases that are involved in psychiatric, in the field of psychiatric. And bipolar disease you see here. And this Timothy syndrome, I think, which is autism. And so CACNA1C, a brief background about that, it codes for the alpha subunits of the L-type voltage activated calcium channel. And it's changed the excitability of the cell if you have mutations in this gene. And it has been linked to patients with schizophrenia and bipolar disease. And here, yeah, I told you about this Timothy syndrome, which is a form of autism. And there it has also been linked to a mutation in CACNA1C. So we wanted to look into this gene a little bit more in detail. And first we started, since I have this background in physics, I had a PhD student there, Erik Smedle, that I told you about. He also came from the field of physics. So we made up a mathematical model to see if we could, you know, understand how the voltage-gated calcium channels, together with other calcium transporters and so on, if they could start these calcium oscillations that we saw, this activity that you saw in the earlier movie that I showed you. And this is a movie again showing these progenitor cells and how they are active. And in this case, in this movie here, we put on a drug called octanol, and that octanol, that drug blocks gap junctions. And what this experiment was important for us to tell us was that there is pacemaker cells in these colonies of cells. And these are cells that are kind of driving this whole activity. So our hypothesis is that this activity that you see in all cells are actually built up by a few cells that are very active. And those cells are triggering networks. So almost all cells becomes active. And when we block this network, we can identify which cells are actually these pacemaker cells. So I will see here, when we add this octanol, we block the network, and then some of the cells are still active. And we wanted to pinpoint and understand those cells. You see here, they are blinking still. So in this mathematical model, we saw that the number of these CACNA1C channels, or voltage-dependent channels, could determine if a cell become active. So in this mathematical simulation, we added this value 20, which kind of mimics the number of voltage-dependent calcium channels. And if we have a higher number here, we get activity. And you see if we have a lower number, then the activity stops. So that could indicate that the number of these CACNA1 channels that I showed you about earlier that we found was actually the one responsible of starting this network or this activity. So the mathematical model could actually predict or mimicked very well if we looked at also with the PCR experiment at different days here. And this figure here just shows you the path that cells should go if they are starting to be spontaneously active. And you see this path going here in the real experiment with real cells. It starts here and then it falls down here. And this is the same here because this lower surface tells you where we have this spontaneous activity. And doing experiments, we could analyze this further. So here again you see you have 4 different cells. And if we add this octanol, you can block out the activity. But some cells are still a little bit active, which then could be these pacemaker cells that we have characterized. And the number of pacemaker cells in our experiments was about 20% or 19%. So if we put these numbers into our mathematical model, we wanted to test the mathematical model if it could actually predict the real world. And doing so, we saw that we got actually the same kind of numbers. You see here 20%, and this is also at day 8. And in this real experiment, we have 19%. So the mathematical model fairly good tells us what is going on in real cells. We could also use electrophysiology to try to do this on a single cell level. And in this experiment, now we looked at the electrical activity. So you see you have these cells are firing with electrical activity. So this is a pacemaker cell. But if we then apply a current that is negative to this cell, this means that we kind of we turn off this voltage-dependent calcium channel. And then this activity goes away. And the opposite, if we take a non-pacemaker cell and then we add a current, we can make them active. So it seems like this number of KGRI1C channels could critically determine the activity of the cell. And that could be very important for these children with autism, and meaning that if they have a problem with this channel, that could inhibit the activity that is very important during development to create like a normal brain. So in the next experiment, what we did was that we developed a KGRI1C knockout mouse. So in this mouse, we knocked KGRI1C out, and we used a Cree EMX animal. And EMX is a gene or protein that is expressed in the developing telencephalon. So it's in the forebrain, and that is a very important region of the brain, of course, for these diseases. And then what we did was that we ran these behavioral tests. And one is called the open field. Another one here is called the elevate plus maze. So this cartoon here illustrates these experiments. So the open field is just an open field like this. And then we have a CCD camera recording. And then we put the animal inside there. And then you can see how the animal is running. And a normal animal should then not run run a lot in the beginning, I think, yeah, and then it starts to rest after a while because it tries to explore the environment. While if you have an animal that is similar to an anxious animal, then it becomes very afraid if it comes into a new environment. So it starts to walk around the walls of this open field. And then after a while, when it feels a little bit more confident, then it can start to explore, and then they start to move more. And what we saw with this experiment was then that this knockout mouse, which is red here, you see that it was actually afraid in the beginning, stayed at the edges of this open field, and then after a while it went out. this was also the same for this elevated plus mace experiment. And we also performed MRI experiments, which is magnetic resonance imaging. And the reason why, this just tells you that it was the same for this elevated plus mace experiment. The reason why we choose to use this MRI experiment was that we wanted to correlate these experiments with the data from this experiment with human experiments, since there is a lot of patient data recorded from patients that have these psychiatric diseases. But there is not so much, with immunostaining and so on that we are usually using in basic science. So we have a small PET core facility at Karolinska. So we left these animals in this facility, and then they could, analyze different regions of the brain. And what we saw there was that these regions seems to be, higher or smaller or larger in these animals that had this CACNA1C knockout. And we looked also for correlation. We did this correlation analysis with all these parameters. And then we found a good correlation here with this mammillary body. Unfortunately, the difference then between like the normal animals and the knockout animals was not significant in this case. But this is our ongoing experiments now that we are trying to analyze. And what we were also looking into, of course, was this calcium activity. So we looked into the calcium activity in this CACNA1 knockout mouse. And what we saw, we analyzed like both frequency and amplitude of these oscillations. And what we saw now was that it seems like the amplitude was actually reduced in these animals rather than the frequency and activity. But this, again, we are still ongoing, and we're trying to analyze this further. So these experiments here, just these conclusions shows that the CACNA1C knockout animal has a lower, it has lower amplitude compared to the wild type. So to conclude the talk here then was that the first story I told you about was this whole tissue biopsy phenotyping of three-dimensional tumors reveal patterns of cancer heterogeneity. And this was a story telling you about how we developed this DIPCO pipeline for analyzing human FFPE samples. And then the other one was about this CACNA1C and how it's involved in the spontaneous activity of progenitors in the developing brain. And at last, I would like to acknowledge the people that have worked on this project and helped us. Of course, Nobuyuki Tanaka and Shigeaki Kanatani, both here from Keio, was very important for the first study. And then Erik Smedler was the first author of the second story. And then, of course, all the other people that have helped us in my lab and also collaborators outside my lab. And also the funders of this project, of course, I would like to acknowledge. So thank you for your attention.
Thank you very much. So any question from the audience? So can I start?
Yeah.
So you said the current can transform the non-pacemaker to pacemaker cells,
Yes.
right? By increase the number of the...
So according to our mathematical model, the number of voltage-dependent channels in a cell determines if it's spontaneous active or not. So if you reach a certain threshold, then the cells become active.
Okay.
So what we did in that experiment was that we tried to test if our mathematical model was true, and so we first we performed a calcium experiment, and we could see what cells were active. And then we put on octanol, and then we saw that they were silent. And then we could patch that cell. And then we could apply a current and get it to become active again.
I got you.
Yeah.
Actually, so in my group, we're working using end microscope or ND star system. And so the deep brain imaging, calcium imaging.
Yeah.
So, for example, so maybe so calcium imaging mammary body. So yeah, it's feasible.
Yeah, that would be, of course, something that would be very interesting to do. But we don't have that equipment to do. This is this that you stick in this.
Downstairs we have.
You have that one. Is it? Okay.
So how deep you can get into?
Yes, in the microscope.
Oh.
Yeah, I know
Very
that there
clever.
is a company from France or something that maybe there are others now that is like you say with an endoscope. You can push it into the tissue, and then where it comes, you record from it. So it's a very powerful tool, of course. And that would be very interesting to do.
Yeah, so undergraduate student working. So.
But you are working with the monkey model with that.
And also on the mouse.
Yeah, mouse you can do it also. Oh, that is
Only
interesting.
2 grams, it's a camera, and so even the free moving mouse is possible.
Yeah, okay.
It's a company called. It's a company in California.
Yeah, that it sounds very.
Inscopics.
Inscope. I have to look into that. That sounds very interesting for us also to use that.
Because there's some findings that MRI volume changes.
Yeah.
I'm very curious what's happening. So calcium imaging, so those areas.
In those areas, and those are of course very difficult to do experiment in intact brains. Usually you do cutting and so on, and that is something completely different. Yeah.
Anything else? Yes.
I'm very interested in the light sheet microscopy. You observed so precise structures. Please tell me the thickness of the light sheet.
Yeah. So of the light sheet, it's how thick? I think it's like 500 nanometers. Yeah. And in this experiment that I showed you, we could actually put in 10 millimeter, a cube 10 millimeter times 10 millimeter times 10 millimeter in this microscope. But usually these human samples that we looked at in this project, they are about between maybe 1 to 3 millimeters. And that is typically also what the pathologists are cutting there, when they do the storing. So we could look through almost all their samples, the entire tissue, with this method. But we had this column. Are you familiar with the column light sheet microscope that was developed by Carl Deiseroth in Stanford? So we built an exact copy of that one in Karolinska that we used for these experiments.
You choose depot. Now, why you choose depot, not scale or cube?
Yeah, you mean iDisco? Why we used iDisco? Yeah. So no, the reason why we picked iDisco was that one was optimized for immunostaining. And of course we wanted to immunostain, but it's also that we wanted to do this re-embedding. And with the iDisco clearing protocol, we are first removing water, and then so it seems to us to be the best and kindest method for the tissue in order to save the tissue afterwards.
Yes.
I'm just interested in the cycle that you have with the re-embedding. Did you notice any shrinkage with the tissue? And secondly, with iDisco, I've noticed it turns into like a rock-hard crystal.
Yeah.
How manipulable is it for pathologists afterwards?
No, but yeah, so the first there was some change of, like you mentioned, Simon was in our lab and worked with this, and you probably saw this, that there are, and that is a common problem with all of these protocols, that you get like a deformation of the tissue. It can shrink, or it can create something. We saw some changes, but we tried to show that in this picture, that the staining was still the same, and it looked like we had a minimal, we affected the tissue in a minimal way, I think. And this rock hard that you told, this is changed. If we use this protocol and go backwards, we can take it backward, actually, through it. Yeah. So and that we could do with the iDisco. I'm not sure if you, was it rock hard, was this with the clarity or scale or?
iDisco.
iDisco, yeah. No, but we can go backwards there. Yeah.
So you said the radius of vessel can predict the cancer stage much better than PSA. And how about if you compare to the classical pathological grade?
Yeah. So what we did then was, of course, the thickness of the vessels cannot be measured with a two-dimensional way. And I don't think that they are looking at the vessels, the pathologists today. They look maybe just at the staining. They can measure this CD34 density just. What we could do then was the picture that I showed you was, since we used samples from the clinic, they were staged already, and we also knew, what was the in some case we had survival data from the patients also. So we could stain it much more. I think that, maybe I didn't stress that, but in this table that I showed you,
this one, yes.
Yeah, so I think that here, if one looks at this CD34 density in 2D, then the rate was this number. While we could have it in this value.
But there is a heterogeneity, in terms of the radius of the vessel.
So this is part of these, that skewness, variance, density, tortuosity, like this. And this is another way. This tortuosity, that is the way, the length. If you have a vessel going from A to B, it could take a straight line.
Parameter.
This is a multi-parameter analysis then. And the tortuosity tells us, how complicated path it takes between 2 points. And this is also classical and characteristic for a tumor, that the vessels are not going in perfect, as good as in healthy tissue, in normal tissue.
How long does it take to prepare the sample?
Yeah, the whole process I think took like 2 days.
2 days.
And then the scanning was like 3-4 hours or something, and then the analysis, but also the immunostaining. It depends on how many antibodies and so on you are using.
So you got a sample from the biopsy or surgical operation?
Yeah, from surgical operation. So we got all kind of samples. I think we have more than 70 samples or something in this study. So we took both biopsies and also surgery. When they did this cystectomy, they removed the whole bladder, and then we collaborated. We had 25 authors of this paper. Most of them are pathologists, and then they went into the bladder and picked up, good regions for us.
Thank you. Anything else?
So the MR-based analysis, so there's no cut mouse.
Yeah.
You show the volume change.
Yeah, of the whole brain. Yeah.
But what it tests is a gold signal and it's fMRI. So maybe so.
No, we have not. No. Yeah, that is an interesting question. Yeah.
And that can be done even for the mouse. Okay.
Yeah.
We published this year scientific reports about using resting state fMRI for the mouse and correlation map. Non-functional correlation map in the mouse system.
Okay, I will check if our facility can do a functional MRI. Yeah, but that is amazing. Yeah, I think in this analysis we had more 114 different values that was measured with these experiments that we tried to correlate with each other. But we are looking into this even deeper now, you know, how we can. But I think it's good to apply, as I said, these methods that are applied in the hospitals, also on animals, you know, because then we can correlate patient data with. So that is very interesting.
Anything else? Yes, Scott. Yeah, thank you. I think you are definitely the key person for our collaboration between Keio and Karolinska, since you have 2 postdocs from Keio and took care of Simon's short visit. And I hope we could continue this collaboration and further develop.
And I will meet also, you know, talk with Dr. Oya here on Tuesday, and so I have a lot of.
Great.
Yeah.
Thank you very much.
Thank you for having me. Thank you.